Yeah.
Okay. All right. Well, happy Tuesday, everyone. Welcome back. I hope you had a wonderful reading week. It is lovely to see you all for what is going to be a wonderful seminar. We're so excited to have Dr. Kiyoko Gotanda. But before I introduce her, I just wanted to say, as everybody knows, we're heading into cold and flu season. COVID is kind of back, and so I do have masks if anybody wants. It's not mandatory, but I'm actually following the lead of one of my personal heroes. I know I told you guys I was going to see Naomi Klein speak in Toronto. It was wonderful, but she was suggesting that the audience mask just to help keep each other safe. So they're here if you'd like them, and I think that'll be ongoing for the rest of cold and flu season. Okay, so now on to the main event, which is so exciting. We are thrilled to be joined today by Dr. Kiyoko Gotanda, who is an assistant professor here at Brock in the Biological Sciences Department. She is also affiliated with us, which we're very grateful for, the Environmental Sustainability Research Center, and also with the University of San Francisco, Quito in Ecuador. Previously, Dr. Gotanda was a postdoctoral fellow at the University of Sherbrooke and a Banting and an FQRNT postdoctoral fellow at the University of Cambridge. Her research lies at the intersection of ecology, evolution, and behavior. And primarily, Dr. Gotanda focuses on two questions, asking about the origins, evolution, and maintenance of biodiversity. Question number one, what are the characteristics of selection? And question number two, can we predict how organisms will adapt and evolve? Dr. Gotanda's research program focuses on a variety of taxa, including Darwin's finches, Trinidadian guppies, and three-spined stickleback. She's also worked with anoles, I might be mispronouncing, parrotfish, and East African cichlids. And so we're so grateful to have you here today. Welcome.
Thank you so much, Jessica. Thank you, huge thank you to Jessica and Marilyn for inviting me to be part of this. I'd like to start by acknowledging the land on which we work is the unceded and traditional territory of the Haudenosaunee and Anishinaabes peoples, many of whom continue to live and work here today, and that the territory is covered by the Upper Canada Treaties and is within the land protected by the Dish with One Spoon Wampum Agreement. So, as Dr. Blythe mentioned, I'm trained in evolution and ecology. And you might be thinking, well, what role does ecology and evolution have in environmental sustainability? And so what I'm going to hope to show you is how some of these underlying principles of adaptation and evolution can be affected by humans, and how that this can have implications when you're thinking about things like environmental sustainability. And so one of the prevailing questions that we have in biology is, why do we have the biodiversity that we have on Earth today? Where did it come from? What generated it? What maintains it? How do we stop it from being lost? And one of the things that I'm interested in, so this is a sort of basically like a tree of life, basically, to show the diversity that exists on the planet. And one of the things that I'm interested in is how the process of adaptation can lead to divergence within and among populations, and might be one of the sources of that diversification in the biodiversity that we have. So a classic example of this idea of adaptation leading to divergence within and among populations are the Darwin's finches from the Galapagos Islands. So what you're looking at here is several species. We know that there's about 18 species that have evolved from a common ancestor that came over from the mainland to the Galapagos Islands. And each of the species has a different size and shaped bill. And so this means that each of the species of finches can exploit a different, what we call a niche, or basically they can eat different types of foods. So on the left side, you have your seed crushers. So these beaks are sort of triangular and designed to crush seeds. On the bottom, you have your more insectivorous birds. And on the right, you have your longer, thinner beaks, which are more for probing. So each of the species has a different ecological niche that they occupy based on these different size and shapes of beaks. So how did this come about? Well, I'm going to walk you through a schematic of how this idea of selection and adaptation can lead to a shift in a trait, for example. So if we go out into nature and we measure a phenotype or a trait, so we were just talking about the finches, so let's say beak size. So we go out there, we catch some birds, we measure their beaks, and then we get a distribution of the trait size or of this particular beak size. So the question is, well, why that particular distribution? Why that mean? Why that much variation around the mean? Why do we have that distribution of these particular phenotypes? Well, what we can do is we can conceptualize what we call a fitness function. So fitness, in a nutshell, is your probability and your ability to survive and reproduce. So if you're this particular beak size, it means you have the highest probability of surviving and reproducing and passing on your genes. If you're a larger beak size or a smaller beak size, you have less fitness, which means you are less likely to survive and reproduce. Now, what can happen is there can be a shift in the environment. Maybe, as Peter and Rosemary Grant found with the Darwin finches, there's a drought, let's say. And so this is going to change the environment and things like resource availability. And so what this does is it shifts this, what we call the fitness function. And so the size, the individuals that had this particular beak size that used to have the highest probability of surviving and reproducing now have very low probability of surviving and reproducing. And these organisms over here that had the larger beak size, now they actually have a little bit more fitness, which means that these larger beak-sized birds are going to have a slightly higher probability of surviving and reproducing. And so this means that we have what we call selection now acting on this particular population of this particular population of birds, for example. And so that's where that idea of one of the questions that Jessica mentioned, what are the characteristics of selection? So what is it exactly what is that selective pressure on it? So in this particular case, in this example, it's drought and resource availability, but it can be other types of things, which we'll see throughout the talk. And we'll go over this too, but you can also get a sense that selection can vary in its strength and also in its direction. And so we'll talk about that a little bit. But right now, because of this environmental change, there's been a shift in the fitness function. So now selection is acting on this population. And if we know these characteristics of selection, what that selective pressure is, how strong it is, and what direction it is, hypothetically, we should be able to predict how organisms will respond. So after several generations, what we would expect is that there will be a shift in the distribution of phenotypes. And so now these individuals over here now again have, with these larger beak sizes, have this higher fitness, so this higher probability of surviving and reproducing. But you can see that compared to where it was, there's been a shift in this beak size. So these kinds of processes can lead to divergence among populations. And if we have this idea of the strength and direction of selection and the causes of selection, we might be able to predict how organisms will adapt to these particular selective pressures. And so to conceptualize a little bit more about this idea of strength and direction of selection, I'm going to walk you through an example. So for this particular case, we're going to look at a trait called clutch size, and that's the number of eggs that a bird lays in its nest. And on the y-axis, you have, we call them selection estimates, basically. Like a lot of things, you're estimating things because you can't get a true mean of a population and stuff like that. So we're estimating selection, and on the y-axis, you can see that there's positive selection, and there's selection that goes by 0, and that there's negative selection. And then on the x-axis is time, which we'll get to in a second. But to conceptualize this idea of fitness, or selection, when we quantify or estimate selection, what we do is we take a fitness trait, so some trait related to survival and reproduction, and we regress it against the trait. So in this particular case, let's say our fitness is fledgling success. So the number of birds that successfully fly away and leave the nest and basically are adults. Now we can have positive selection where your trait, your clutch size, the more eggs you lay, the more fledglings you're going to have. So this is positive selection for having more eggs or having a larger clutch size. So this might be a situation where maybe there's lots of resources. It's a really great year, there's lots of invertebrates and there's lots of caterpillars, and the mothers and the fathers are able to feed all the young, and they all fledge, and they have lots and lots of young. So they have high fitness because they laid a lot of eggs. In other situations, you might have where it doesn't really matter how many eggs you lay. You're going to get the same number of fledglings. So there's no selection on the number of eggs for a female to lay. But then you can also have negative selection, where the more eggs you lay, the less fledglings you'll actually have. So this might be a situation where there's not a lot of resources. And so, there's situations where you've got 10 offspring to feed, and you really can only afford to feed 2. That means you've actually lowered the number of fledglings that you're going to have because you laid so many eggs and you don't have enough resources to provision for all of them. Whereas if maybe you'd only laid like 3 eggs, you would have had 3 fledglings. So there you can have what we would call negative selection. So when we consider selection, historically what we've done is we've gone out, we went to a population, we looked at a fitness measure, we looked at a trait, and we estimated selection. Now, if we do this at one point in time, this makes the assumption that selection is consistent through time. However, if we look at actual data, so these are lovely little birds called great tits that are very common over in Europe. And if you look at actual data, you can see that actually this selection can be highly variable through time. So some years, it's better to have lay less eggs or have a smaller clutch size. Other years, it's better to have a larger clutch size. Some years, it doesn't really matter. And from year to year, it varies not only in the direction, so you have positive selection and negative selection, but it can vary in the strength of selection. So in this particular year, selection was stronger than it was on this particular year. So again, if we do this though in one population, we get a sense of the temporal variation in selection. But what about but that makes the assumption it's going to be the same in another population. But if we go out again and look at actual data from a second population, you can see that the selection can be really variable. And what's interesting is that within 1 year, you can have selection that varies in the direction, positive or negative selection. And in other years, you can have selection in the same direction, but the strength of the selection is highly variable. And if we look at more populations, it just gets like, oh my goodness. So previous work had looked at the temporal variation, and my colleagues and I were interested in the spatial characteristics or the spatial variation in selection. So this is work that was done in collaboration with Adam Sapilsky, Mike Morrissey, Sarah Diamond, Joey D'Acusa, and Stephanie Carlson. And so what we did was we compiled a database of selection estimates that were estimated in at least 2 populations. So there's spatial replication, basically. And this database spans all taxa, so plants, vertebrates, and invertebrates, and it has a global distribution. And so we wanted to know if that spatial variation in selection varied more in the strength or in the direction of selection. And what we found was that the selection tended to vary more in the strength of selection as opposed to the direction. So selection is pushing on a population in a certain direction, in the same direction across 2 populations or more, but it's more, the strength of that selection is what's varying. And then we looked at also global climate patterns that might be driving these patterns of spatial variation in selection. And interestingly, we found that precipitation is a driver of spatial variation in selection, but not temperature. So we thought that was quite interesting. But there are climate variables that are driving these patterns of selection. So we're getting this idea of this sort of abstract concept of selection. And so what if we take this out and put this into an actual system? And so I'm going to introduce you to the guppy system, the Trinidadian guppy system. They are lovely little freshwater fish. Any aquarium enthusiasts with guppies in their aquariums? No? Goldfish? Siamese betas? No fish people. Okay. So they're sexually dimorphic, so the females are larger, they have no coloration except for this little gravid spot on the belly, whereas the males have all these really pretty color patternings on the side. And if you give a female a choice between a conspicuous or a really brightly colored male and a less colored male, the female is going to prefer the male with more color, right? So there's sexual selection acting on male guppy colors to have more coloration because that means he's going to have a higher probability of surviving and reproducing. Now, if you put the same 2 brightly colored and less brightly colored fish in front of a visual predator, the one that's more brightly colored is going to be seen, and then it's going to be consumed. And so that means you have natural selection for lower coloration. So the guppy system is this really nice interaction between sexual selection for more color, whereas there's natural selection for less color acting on the same system. And the reason we know that predation is a really strong driver of this is because of this system. So this is the north coast of Trinidad. There's a really beautiful mountain range up here. Trinidad is just off the coast of Venezuela. It used to be part of the mainland, and then due to geological time, it's now an island. And on this northern mountain range, you have these repeated river systems that have these beautiful waterfalls there. And these waterfalls basically act as natural barriers to predators. So if we look above the waterfall, there'll be a what we call a low predation area. So there's guppies up there, And they coexist with this killifish, and the killifish don't really eat the guppies, so there's no natural selection for them. There's just sexual selection going on in these populations. And so these populations, the males are really colorful because there's no predation there. If you go down below the waterfall into these high predation areas where you've got these visual predators, the populations have a lot less color because you've got that natural selection acting on the male guppy color. And what's really cool, the other reason that we know that predation is a strong driver is that a lot of researchers have gone in and taken guppies from the high predation area, went and found some area above a waterfall that didn't have guppies in there before, took them, put them up there, and then within a couple of generations, the formerly drab, low color guppies from the high predation area, you move them up into a low predation area, within a couple of generations, the population has more color. So we know that this natural selection against color is a strong driver of variation in male guppy color in these particular populations. Now the question is that kind of selection consistent through time? So in collaboration with Andrew Hendry, what we did was we looked at different populations of these high below the waterfall and low predation guppies, and we looked at their color over 6 years. And what we found was that in blue are your low predation areas, so that's where there's no predators, but you have sexual selection for more color. And then we looked at two populations of high predation areas where you have your predators, and so you have a natural selection against high color. And the take-home message from this is that the high predation sites, for the most part, generally do have lower color due to this natural selection against color, as compared to their low predation counterparts, and that this difference is relatively consistent through time. There is a general uptick in color, and we think that's due to our photographic methods. Back in 2005, we used early generation digital photographs, and by 2009 and 2010, we were using digital SLRs with ring flashes. So we think it might have to do with the quality of the photographic equipment. But the main thing is that this is showing that predation is relatively consistently a strong driver of differences in male guppy color in the wild. So if we know that predation is the strong driver of selection, then hypothetically we could predict what will happen. And so I mentioned to you how, so this is work done in collaboration with Amy Pack, Carolyn LeBon, and Andrew Hendry. And as I mentioned before, what researchers have done was they have taken high predation guppies, put them into a low predation environment, and they evolved more color. So that was done in the wild, and it was also done in the laboratory. And so what you're looking at here is a mesocosm experiment, so enormous tanks, and John Endler took high predation guppies, mixed them up a whole bunch of times. So he took 18 populations of guppies and just kept breeding them and mixing them up to maximize the genetic variation. And then he started several mesocosms with 200 guppies. And then within 2 years, a year and a half, there was an increase in color. And so again, that's showing that taking away that predation, that selection, the natural selection, the population will increase in color. Now, as I mentioned, he did this though with starting populations of 18 guppies, and he mixed them up to maximize genetic variation, which from an evolutionary experimental point of view is fantastic. But it's not representative of what would happen in nature. In nature, most likely you're going to get maybe one pregnant, so they're internally fertilized, so the females can have multiple male sperm storage, and if the pregnant guppy accidentally gets up there by a kingfisher flying it up there, you're not starting with 200 fish from 18 populations. You're starting with a handful of fish from one particular population. And so what we wanted to do is we wanted to emulate this experiment, but with sort of more natural, realistic conditions. So we used two source populations, the Rico River and the Gloria River, and they're from two drainages, and these drainages have been separated for about 1 million years. So these two rivers have different evolutionary histories. And so we had two source populations, and then we did a mixed population where we put fish from both populations. And we did this in these giant mesocosms, and we replicated them 3 times each. And so to give you a sense of what a mesocosm is, we've got these 300-gallon cattle troughs And we painted them, put gravel in them. This is what the guppies got to live for a couple of years. And then every 4 months, we went in and we fished out all the guppies. We took some scale samples, which I actually still have. I haven't done anything with them. And we took photographs of the guppies. And so I'd like to give a shout out to the only technicians I could afford, which were my partner's niece and nephew. But actually, no, that's kidding. They really had a lot of fun time though making liver paste, which is one of the foods that we make for the fish, which entails blending livers and then mixing it with baby food and then cooking it. So they had a very grand time. But in reality, I do want to give a thank you to everybody who helped me both with the maintenance and running of the experiments as well as with the processing. And as you can see, a lot of these experiments, it takes an army or it takes a village to get it to happen. So I'm very grateful for all the support that I received for this. So I'm going to show you what we call biologically relevant color categories with Trinidadian guppies, because we quantify 8 colors and putting up 8 plots would just be, whatever. So we grouped them into biologically relevant categories. So we have our carotenoids, which includes our orange, yellow, reds, and bronzes, and these are generally dietary based. We have our structural colorations, and so these are the reflective colors, so the blues, the silvers, and the violets. And then we have our black colorations, which is usually produced by melanins, and these are what we call a black and a fuzzy black. Okay, so I'm only going to show you month 0 and month 12, and 1 year is enough time for there to have, and previous work has seen a shift in male guppy color if you've taken high predation guppy and moved them into that low predation environment. And so that's basically what we did, was we took high predation guppies, we took 10 males, 10 females from those 2 different rivers, and then we mixed them up as well, put them in a mesocosm and said, have a good life. And so what we were predicting based on previous work was that there would be an increase in the carotenoids and the structural colorations, because these are, with the carotenoids being dietary based, they're like an honest signal. So if you've got a lot of orange and you're really brightly colored with orange, you're telling the female, hey, I know how to find food and I'm good at it. And so that's a good attitude to have if you want your offspring to survive and reproduce. And then the structural colorations are also involved in sexual selection, we think partly due to things like contrast and stuff like that. And so we predicted that there would be an increase in the carotenoids and the structural colorations, and not much has been found with the melanics in previous research. And so what we found, and we also thought that because of the different evolutionary histories, we might expect to see the mesocosms, the 3 replicates acting in sort of the same direction, if you will, because of this shared evolutionary history. And what you can see is that we didn't find that at all. So you can see that some groups increased in color, in orange color, some decreased. And then if you look at the different colors, they're not going in the same direction at all. It's all sort of idiosyncratic. And we also found this with the structural coloration. So some mesocosms decreased in structural coloration, some increased. There wasn't really anything going on with within a population. And then what we did find, though, interestingly, was that there was an increase in the melanics. It was relatively consistent. And so we think there might be a couple of different reasons. We think it might be due to variation in sexual selection. So females in general prefer more color, but there's a lot of variation in just how much they prefer that color. And if you start a population with only 10 females, variation in that preference for color can be amplified. So we might have just happened to pick more females that preferred more blacks. But if we'd started it with maybe 40 females, we might have seen something different. Another possibility is we did, so I forgot to tell you, those mesocosms, we were in a pig farm, basically. So it's just this big, massive barn, and we had to use water heaters to keep the temperatures up. And so we actually were keeping them at a couple of degrees lower than what is standard for keeping them in enclosed areas. So there might be a thermoregulatory aspect to the increase in black coloration. So with these guppy systems, we understand that predation is a strong driver of selection, and we try to predict how organisms were going to adapt, and it was very different. But this is those are in natural populations. In my talk, I was going to talk about human influences, and I haven't really said anything about that yet. So we have been inundated over the past, 5-10 years with these really sensationalist headlines about how things are declining and we're in a mass extinction and, we're losing our biodiversity. And I know some of these studies underlying these headlines are controversial, But the main point is that what underlies all of these titles is that they deal with us humans, that we are a contributing factor to this in some way or another. And for better or for worse, Palumbi back almost 25 years ago wrote this paper that was basically like humans can be considered one of the world's greatest evolutionary forces. And that's because we're driving a lot of those patterns of selection, a lot of those selective pressures. It's us. We're driving them. And so my colleagues and I, we put together a special issue that focused on all these different types of human influences. And this is not an exhaustive list. This is just scratching the surface. But I'm going to talk to you about 3 of them today. Eutrophication, urbanization, and invasions. And I'm going to do it through asking 3 questions. So the first is, does dissolved oxygen levels affect behavior and swimming performance in East African cichlid? And this work was done in natural populations. So naturally, populations have varied naturally in their dissolved oxygen level, but it has implications in terms of human influences because things like eutrophication due to pollution can alter those dissolved oxygen levels in water. So understanding how organisms might or might not adapt to different levels of dissolved oxygen, we can start in a natural system and then extrapolate from that into human influences. So first, what is it about dissolved oxygen? So this is work that was done in collaboration with Aaron Reardon and Loren Chapman. So we worked with these East African cichlids. They are called common name is the Egyptian mouth brooder, and mouth brooders are really cool. So what happens is the females, they lay their eggs, and then they take the eggs up in their mouth, and then the males have these egg spots on their anal fins. And so then the female's like, oh, look, there's more eggs over there. So they go over there and they try and pick up the eggs from the egg spot, but of course nothing happens. But it stimulates the male to release the sperm. So the sperm then fertilizes the eggs in the mouth. And then the female hangs out for a couple of weeks while the eggs develop into fry. And then when they are old enough, they will swim out of her mouth and do some stuff. And then if there's a predator that comes by, she'll vacuum them back up into her mouth until they are ready to forage on their own. So it's really cool. Fish are really awesome in that sense. They just have some, they're like birds. Like, why are you doing this? Anyways. And what's great about this particular cichlid is they co-occur in areas of both high dissolved oxygen and low dissolved oxygen. And just like us, they need oxygen to survive and breathe through their gills, basically. So in these open riverine areas, you have high levels of dissolved oxygen. And then in these dense papyrus swamps, you have areas of low dissolved oxygen. And so what we did was we took fish from low dissolved oxygen, we brought them back into the lab, we split them into 2 groups, and then we acclimated them to 2 different levels of dissolved oxygen, one high and one low. And so you've got the same population of fish, and they're acclimated for 5 months to these different oxygen levels, basically. And oxygen is important for behaviors because it's an energy source, right? Or it's part of the generation of energy. And so what we did was we looked at different types of behaviors. So they have these aggressive behaviors where they do these like open mouth charges at each other, the males do when they're fighting. And then they have these mating behaviors. And so the males do this little like wiggle thing in front of the females. So we looked and quantified both of those behaviors in the high dissolved acclimated fish and the low dissolved acclimated fish. So again, these fish are from the same population. They've been acclimated to 2 different oxygen conditions for 5 months. And they engaged in many more aggressive behaviors than in their mating behaviors. But in the high dissolved acclimated fish, they had significantly more displays. So this is showing that even though they're coming from the same population, you let them acclimate to a high dissolved oxygen environment, they're going to engage in more displays, hypothetically because they have more energy to engage in these kinds of displays, because they have more oxygen available to them. So we know that oxygen can be an important factor in differences among individuals. So can we use this information, again, to predict how they might adapt to these different types of environments? And so this is work done with Aaron Reardon, Sean Murphy, and Loren Chapman. And so what we did was we went out, we collected fish this time from the field, from both high dissolved oxygen populations and low dissolved oxygen populations. We brought them back up to the lab, we reared them under the same conditions they were and allowed them to reproduce 1 generation. And when we look at the morphology of them, these low dissolved oxygen adapted fish, they've got larger gills, which makes sense. You've got more surface area for diffusion of oxygen. And if you're in a low dissolved oxygen, so it's similar to people who grow up in upper elevation areas, right? Their lungs are different than those of us who grow in terrestrial areas because of the amount of oxygen that's available. And coupled with these different sized gills are fatter heads and deeper bodies. But the question is, does it make sense for larger gills to have more oxygen? But then why don't the high dissolved oxygen weird fish have larger gills? Because they'd be like superheroes, right? If they have these larger gills and are in these high oxygen areas. So why don't they have these larger gills? So these high dissolved oxygen fish have smaller gills, thinner heads, and shallow bodies. Now, what I predicted was that it would have to do with their swimming performance. Because when you've got different, fatter heads and deeper bodies, and thinner heads and shallower bodies, you would predict that the swimming performance would differ. So if you think of like a tuna versus like a flounder or a halibut, right, their swimming performance is very different based on their body size and shape. So we thought that there was going to be a difference in their swimming performance. So we looked at what we call a fast start response. So this is the fish. This is an artificial stimulus. And what we did was you basically use a stimulus to startle the fish, and it bends into that little C shape, and then it swims off. So it's kind of escape response, basically. And we use the high-speed video and analyze it frame by frame, and you can get different characteristics like maximal velocity or maximal acceleration, and those sorts of metrics of swimming performance. And we didn't find any difference. They're the same swimming performance. And so you're kind of like, oh, dang, what's going on here, right? So, we're like, they've got different shaped bodies. Why are they having the same swimming performance? So we delved a bit deeper and we looked into what we call reaction type. So when they make that response, that startle response, they can either bend once into that C shape and then they'll swim out that way, or they can engage in what we call a double bend response. So they'll bend into that C, they'll bend the other way, sorry, my fingers don't go that way, before it'll start to swim off. So basically the fish can bend once or bend twice as its startle response to a particular stimulus. And we know that double bend responses elicit higher maximal velocity and acceleration. So if you engage in a double bend, you're going to go faster than if you engage in a single bend. And when we looked at what types of reactions the fish had, we found that those reared in the low dissolved rearing treatment, so those with the deeper bodies and bigger heads, they almost always engaged in a double bend. Whereas the high dissolved rearing treatments, it was about 50-50 which type of bend they engaged in. So this means that what we think is going on is that these low dissolved reared fish are engaging in a double bend, and we think that that double bend might be more energetically costly. So there is a cost, potential cost that they have to engage in this double bend response to be able to achieve the same type of swimming performance as their high dissolved oxygen reared counterparts. So does dissolved oxygen levels affect behavior and swimming performance? Yes, it does. So next I want to take you to the Galapagos Islands to ask about Darwin's finches and their response to the presence of invasive species. So the Galapagos Islands are an island archipelago. They're located about 1,000 kilometers off the coast of Ecuador, off of South America. And they're famous for a lot of things, but one of the things they're famous for is they're quite often featured in all those wonderful documentaries, Planet Earth 2, Blue Planet, and all those sorts of things. So there's a famous clip from Planet Earth 2 that's won a BAFTA, the UK equivalent of an Emmy, but there's a new version out with a different narration, so.
Oh, there we go. Little cute little lizard. What the hell is going on? He's being watched. Snake's a straight asshole. You can tell the way they're looking at it. They plotting right now. See how they got their necks up? That means they coming to get it. The lizard don't even see what's happening. But you understand me? One thing about a lizard, I think he had Geico. If he had Geico, he got a 15% chance of making it up out of here. He beat Gary right now. He's saying it. He's saying it. You watch. Oh, get out of there, man. Go. Did you see the feet on me? Oh, they coming from everywhere, cuz. This is crazy. Oh, that's up. You were just richer than the snake bill USA. There's snakes coming from all angles. As he dips and dashes, he makes a getaway, jumps into the arms of a big snake to twist his arm, but they twist it themselves. Find the loophole. And you got to find your way to the top of the mountain on that dude. He's jumping. He's very well trained in escape tactics, as you can see. Oh, good move. I like that. I like that. Oh, jump. There you go. Get a hold of it. See, snakes can't do all that because they ain't got heads. And they ain't got feet. Ooh. Yeah, because I'm up on the top of the mountain with you. Remind me of my homeboys when they run from the police. Clean getaway, man.
All right. And apparently, Sir David Attenborough, who did the original narration, approves of this narration. So there you go. So the Galapagos Islands are also famous for where a young Charles Darwin visited on his voyage of the Beagle, which is shown here. And he documented his travels around the world in a really lovely book called The Voyage of the Beagle. And this is a photo of a first edition. When I was over at the University of Cambridge, I found out our zoology library had a first edition. So it's like, can you get it out? And I'm like, it's so cool. And you can look at all the plates and everything. It's really beautiful. So Darwin documented what he did as a naturalist in this book. And this, on the Galapagos, this included doing things like riding the giant tortoises and doing things like picking up the marine iguanas and throwing them in the water, and then the marine iguanas would swim back to him and pick them up and throw them in, swim back. And he's just like, okay. So we are not allowed to do that anymore. And for those curious, this is a commissioned bronze sculpture by Felix Dennis, and is part of his Garden of Heroes and Villains, which has over 50 life-size statues of various people and other things. So as we mentioned about the Darwin's finches, which we've seen already, we know that multiple species of Darwin's finches have evolved from a common ancestor through that idea of adaptive divergence and the different selection pressures. And so they now all have this different size and shaped beak. Just as a side note, it's really interesting. These are called Darwin's finches. When Darwin went to the Galapagos, he really didn't care about them. He's like, oh, they're kind of drab color. They don't have a really good song. The mockingbirds are much more interesting. And so he collected the finches, and he taxidermied them, and he labeled them, and he threw them all into a bag. But he didn't note which island they were from. So he got back to England. And he's looking at all this beak variation, and he's like, wait, I remember some of these were like on some islands, but not on other islands. And he's like, okay. Fortunately for him, his fellow colleagues on that journey properly labeled their specimens. And he also had museum specimens. So this is a lesson to you all in your data collection. Always label your stuff with more things than you think are necessary, because you never know what's going to happen. So in terms of Darwin's finches, and in terms of that, what are the characteristics of selection? What is that selection? Well, unfortunately, in the Galapagos Islands, we now have invasive mammalian predators, including cats and rats. And the reason this is a problem is because, that as Darwin noted in The Voyage of the Beagle, all of these terrestrial birds are often approached sufficiently near to be killed with a switch, and sometimes I let myself try with a cap or a hat. And what's important about this is that this means that there's an evolutionary naivete of these land birds to mammalian predators. So these birds, until we came along, had never seen a mammalian predator. So, and as we know, cats and rats can be quite strong predators. And so the question is, well, how will the finches adapt to the presence of these invasive species? And so what I did was an among-island comparison. So 4 of the islands on the Galapagos Islands have the presence of house cats and black rats, and 2 of the islands are pristine. So no history whatsoever of any of the invasive predators. And so what I looked at was a metric called flight initiation distance. So it's the distance at which a prey will run away from an approaching organism. And as you, if you have very low anti-predator behavior, it means you're willing to let that potential predator get very close to you before you make that decision to run away or fly away. As you go up the y-axis, that distance will increase, which means that as a predator approaches, you're like, oh, okay, no, I'm going to leave. And there's going to be a larger distance between you and the approaching predator when you make that decision to run away. So we have a baseline sense of their anti-predator behavior on the pristine islands. And on the islands that have the presence of house cats and black rats, their anti-predator behavior was significantly higher than on the islands. The finches' anti-predator behavior is significantly higher than it was on the pristine islands. One of the things that the park has done, though, in an effort to control these, is they have successfully eradicated the cats and rats on some of the islands. And what's really great about these eradication is that we can figure out if maybe this increase in anti-predator behavior is actually an evolved trait, or if it's just what we call a plastic trait, where it's just due to the environment. So once we remove the predators, that selective pressure for that increased anti-predator behavior is gone. So there's a cost to that increased anti-predator behavior, and the trait's purely dependent on the environment, where I would expect to see a reduction in the anti-predator behavior. But if you remove the predators and that anti-predator behavior is named, that might mean it's a heritable trait that is being passed on through the genes. So that might mean that the trait is evolved. So here are the original data that we found. Here's our 2 islands that had successfully eradicated the house cats and flat cracks. And you can see that the increase in anti-predator behavior has been maintained. So this suggests that this adaptation to the presence of invasive behaviors is an evolved trait in terms of increased anti-predator behavior. The one thing I can't rule out is that there might be cultural transmission, and so that this increased anti-predator behavior is being learned from offspring by offspring to their parents. Or, well, anyways, you know what I mean. They're learned. So there is that possibility, which I can't rule out. And the islands were eradicated in 2003 and 2008, and these data were collected between 2016 and 2018. So enough generations have elapsed that if this was just a trait due purely to the environment, enough time has elapsed that I would have expected to see a reversion to the baseline levels. So the Darwin finches have adapted to the presence of invasive species, and so the last thing I want to look at is urbanization. And urbanization, you might be like, well, okay, urbanization, we all live in cities and that sort of stuff, so what's going on? So to do this, we compiled a database, phenotypic rates of change, evolutionary and ecological database. And in a nutshell, it's a database that looks at a particular trait and if it's changed through time. That's it. So has your body size gotten larger? Has your body size gotten smaller? Has your arm length gotten larger? Has your arm length gotten smaller? Has your metabolism changed? That kind of stuff. And so this database has had a huge number of contributors and people who contributed to it. And like this question database, it's a global database that spans all taxa, so plants, vertebrates, and invertebrates, and it also has a global distribution. And what we were interested in is that we wanted to see how characteristics, different types of human influences might be affecting that trait change. Are there certain types of human influences that might be making the traits change faster or slower? And so what we did was so basically what you have here, so we looked at different human influences, and basically the further over you are this way, the faster you're changing, and the more over here, the slower you're changing. And what we did was we looked at different types of human influences, so pollution, response to introductions, hunting, harvesting, introductions, landscape change, that sort of stuff. And the take-home message here is that for several of these types of human influences, you do have higher rates of trait change. So human influences can affect the amount of trait change that a population is experiencing as compared to when it's not experiencing any type of human influence. I know climate change seems a bit odd with it being over here, but there's various technical reasons for that, including that climate change can have very conflicting methods, and then also you have short-term versus long-term climate change effects, which is not accounted for in this database, which might account for why it seems like there's not a lot going on with climate change. Now, this, you'll notice, does not include urbanization. So using the database in collaboration with Maureen Alberti, Christian Correa, John Marslow, Andrew Henry, Eric Kokovacs, Victoria Hunt, Travis Aspar, and Ethan Ju, we did look at different aspects of urbanization. So we attributed different types of urban disturbances to the trait changes. And basically, you have your natural populations that aren't experiencing urbanization. And you can see that some of these urbanization disturbances have increased the phenotypic rate of change. So again, the further this way you are, the faster your rate of changes. So urbanization appears to be accelerating trait changes. So this means that urbanization can be having an effect on selection and adaptation. So based on this, I'm almost done, I promise. So on 4 of the islands, on the Galapagos Islands, we now have permanent human populations. And so there's been an exponential increase in the number of tourists and the number of human populations. These data go to 2010. The most recent data I have are from 2018. In 2018, there were 280,000 visitors and a permanent human population of 30,000. These numbers, of course, have changed, but I have gone back since the pandemic, and it's still going up, basically. Now, the thing is that us humans can't eat the foods that are already on the Galapagos. We can't eat the foods that the finches eat. So we've brought our own foods there. And what we know, though, is that the types of foods they eat is a strong driver of the different sizes and shapes beaks they have, right? Some eat insects, some eat seeds, some go and eat the center of the nectar from flowers. So we're bringing in our food, and the important thing is that it doesn't matter what size or shape beak you have to be able to consume human foods. Any of the species of finch can consume cooked rice or popcorn, or in this particular case, breakfast right out of your cereal bowl. And have milk on it too. So we wanted to know how are Darwin's finches preferentially consuming human foods. And so this is done with Diana Sharp, Jaime Chavez, Luis Leon, Jeff Porters, Andy Hendry, and Yost Raymakers. So what we did was we presented them with different types of foods, the foods they're supposed to be eating, and different types of human foods. And so this was a cafeteria-style experiment. And so we would put the trays out, and then the finches would come. So this is a trial that's in town. And so clearly the finches are coming, they're eating stuff out of these trays. So the question then becomes, what exactly is it that they are consuming? So we did this at 3 sites, a site with no human presence, a site that has the presence of humans, and these humans bring human foods. There's camping and picnicking here, but it's not an urban area. It's a good 30-minute drive from the urban area. And then we also did this in town. And so I'm going to present to you for each site a plot. And basically, on the left side, you have your human foods. And on your right side, you have your non-human foods, or the foods they're supposed to be eating. I made these slides in England. Crisps translates to potato chips, and biscuits translates to hard cookies, and then rice is still rice. And so what we found was that in the non-human areas, that in general, the finches tended to not come to the trays, and when they did, they just didn't, were ambivalent. But at the sites where there is the presence of human foods, the Darwin's finches will preferentially consume that human food. So on the beach, they are preferentially consuming the human foods, and the same thing in town. So we now know that the Darwin's finches preferentially consume human foods when they are exposed to the presence of human foods. Now, the other thing I want to talk to you about is anti-predator behavior in relation to urbanization. So we can make 2 predictions. In urban areas, you might expect to have increased anti-predator behavior because there's maybe a higher density of predators. Maybe there's more resources. Water is a very limiting resource on the Galapagos Islands, and so there's much more water in the freshwater town area than there will be in rural areas. But also in town, you have a large number of stimuli. So you've got trucks flying around and people and all sorts of stuff. So maybe there's a decrease in anti-predator behavior due to habituation. And so here are the data that I already showed you. The 4 islands that have the presence of house cats and black rats are the same islands that also have the urban areas. These data that I showed you were collected from non-urban remote areas, so not in town. And so the question is, what happens with the anti-predator behavior in town? Well, what I found was that the anti-predator behavior in urban areas is lower compared to their non-urban counterparts. And what's interesting is that this that urbanization can strongly counteract any adaptations to the presence of invasive predators to the point that their anti-predator behavior is lower than what you would find in populations of finches that have no exposure to urbanization at all, or anti-predator behavior. So I do have a confession to make. All these data were collected with either myself or a field assistant as a simulated predator. And I've been talking on about house cats and rats, and you're like, well, you know, Kiyoko, you don't look like a cat, you know, like, what's up with that? And I'm like, okay, that's a fair comment. And so I tried to get a real cat, and I couldn't. So I did the next best thing, and I got a stuffed cat. And you'll notice, this is Bartolome, and you'll notice Bartolome cannot move. So Bartolome got stuck onto a remote control vehicle, and it was used as the stimulus. I had a whole family of cats here, and I named them all after different islands. And so basically, there's a finch up here. And so this is the cat chasing the finch. Finch flies away. And then the beauty of this metric, this flight initiation distance, is you put a marker where the bird was, you put a marker where the stimulus is, and then you just get a measuring tape out and you measure the distance. And that's how you quantify flight initiation distance. So it's a really lovely metric to collect in the field in terms of anti-predator behavior. And if you want to see what the poor finch saw, okay. So for logistical reasons, I can only replicate the urban-non-urban comparison in terms of anti-predator behavior. So on the left is you have the human stimuli, and on the right you have it when we use the cat as a stimuli. And the statistical analysis shows that it doesn't matter the type of stimulus you use, it was more important which environment you're in, whether or not you're in an urban or a non-urban area. So I would argue that even though the data collected with myself we might not look like cats, it's still our interpretations in relation to invasive predators is still valid. So has urbanization influenced adaptation in Darwin's finches? Yes, it has. All right, so I showed you that dissolved oxygen can affect swimming performance and behavior in East African cichlids. I showed you that Darwin's finches have adapted to the presence of invasive species by increasing their anti-predator behavior, and that this increase is maintained even when you remove the predators, suggesting this is an evolved behavior. And I showed you that urbanization can influence Darwin's finches. We know now that Darwin's finches preferentially feed on these human foods, and when given a choice, if they've been exposed to them, and that urbanization can strongly counteract any adaptations to invasive predators in terms of decreased amounts of flight initiation distance. So in terms of us humans having an effect on how organisms are adapting to things, for better or for worse, we're here, and so I think that's something that we need to consider. So what I hope I've shown you is that there are the consequences of human interactions with nature, that humans are affecting evolution and adaptation by influence and selection, but also that this idea of ecology and evolution, while it might not seem intuitive to account for in environmental sustainability, you can see now how these types of human influences, we might be affecting other organisms, which will have repercussions on our decisions that we make regarding environmental sustainability. So I apologize for going over. I was just having too much fun showing videos and stuff like that. So I'd like to thank everybody who helped me in the field, all of my funding sources, and I will be happy to take any questions. And I'm happy to stay late too, because I know a lot of you have to go very soon. So I'm happy to stay late to answer any more questions you might have. So.